Oh yeah?
I think so. I feel like it's actually
Feels good, huh?
Feels good.
see even a
Yeah. So it'll be between Guy and Alex. Oh yeah?
Dear colleagues, it is a great honor and a real pleasure for me to open a single molecule localization microscopy symposium 2016. We are all delighted to have you here to participate and to share with us latest progress in super resolution imaging and its successful transition to the biological sciences. We are especially honored to have Dr. Lipikon Schwartz and Dr. Javi with us today, whose keynote lectures will also be open to the public. So we will make a small question for you, if you can just be in this part, because we would like to have public in these areas. Format of this year conference is a little bit changed, because we are hosting as well 2D, 3D software image competition, which is actually focused. This is the second edition of this software challenge, and it is focused on a 3D localization techniques, astigmatism, B-plane, and double helix approaches. We are also very grateful to our generous support from the sponsors, especially our main commercial sponsor, Nikon, Swiss National Science Foundation, EPFL, NCCR in chemical biology, who also helped us to have keynotes open to the public. I will be short to allow a software competition to have a bit more time, because they have squeezed a nice program and a lot of talks in a very short time. And I wish you a very fruitful conference. On behalf of Scientific and Organization Committee, I thank you all for your attention. But just before starting, a few more kind of organizational things. So for if you're not from EPFL or from a university and you don't have an EduRAM, you can connect following these instructions that are also given to you from Helen. Also, if you have a color code on your backside, please contact Helen again. You will know who has it. Then we are also having image competition. You have received during your registration basically popular vote. It will be a popular vote, and we will give a prize at the end of the conference, which is actually a very nice Nikon digital camera. With this, I would like to conclude with a welcome and organizational parts. Maybe from time to time we will jump in with some announcements, but for now, that's it. Thank you.
Do we have some images? Yes. No. So sorry. Sorry, so I will start during we solve this technical problem. So it's a pleasure for me to attend this session. This is a special session on the software localization microscopy. So we have a lot of events, a few events about the software challenge. So first we have this special session for today. At the end we have roundtables. And tomorrow we have also some poster sessions. So during the poster session there is a poster about the software issue. And we have also organized a demo, so some people can make demos. So and next Tuesday we have also the Seamus presentation, which is round up all the challenge issues. So that's the program for today. It's really tight. We have 10 talks, and at the end of the roundtables. So I should say a word that we have video streaming, and the presentation of now is recorded. So it's nice to see a lot of people for the software problems, even if it's a Sunday, very Sunday, but we have a lot of people in the room. That's very nice. Okay. So the first speaker is Hazel Babcock. I can call him, yes, and start this presentation, which is on my computer. Yes. Thank you. Thanks to Hazel Babcock to come from Harvard. So Hazel Babcock participates a lot to this challenge. He has a lot of contribution, and also he participates with 3 softwares. So it's one of the main participants of the challenge. Thank you.
So I'm hoping that a few more I might actually win. So good afternoon, my name is Hazel Babcock, and today I'd like to talk to you about using cubic spines to fit some of the molecular localization microscopy data. Okay, so what is the problem we're trying to solve by using cubic spines? Well, most PSFs, most microscope PSFs are not actually Gaussians. So even though we typically approximate them as such. So, for example, the one on the left is the actual astigmatic PSF for the challenge, for this year's challenge. And then on the right, you see the best fit Gaussian. As you can see, they look different. And my hope is that by using a spline, I'll be able to do a better job of fitting than if I had to just approximate this with the Gaussian. So many groups have actually used polynomial splines before for this purpose or similar purposes. And what we're going to do is, what I'm going to do is polynomial PSF using cubic splines. So in one dimension, they're just third order polynomial, and then they're each, as an example, so each of these polynomials is valid on a short interval. So you have your gray bars in PSF. PSF is upsampled to give you the red dots, And then each of the blue lines is a single one of these polynomials, so they're each valid on this small interval. And then they're also, the coefficients are calculated so that they're continuous at the red dots, and they're also, their first derivatives are continuous. In 3 dimensions, it gets a little bit more complicated. I have a few more terms. But again, it's basically, it is just polynomials. And now you have this polynomial, this 3-dimensional polynomial with 64 coefficients, which represents your spline in the cube rather than on the line. So, a quick summary of what our procedure is for this approach. So, first you need to measure the PSF of your microscope. So you can, for example, take Z-stacks of small fluorescent beads. Then you can slice sub-images of, you want the isolated beads out Z-stack, you align them and you average them to create sort of average Z-stack. And then we're upsampling this by 2x and x and y. And then once we have done that, we then can calculate the spline coefficients using a little bit of linear algebra for each of these little cubes. And then finally, we're going to use this cubic spline to represent the PSF in 3D. And then we're going to use a maximum likelihood estimation fitting approach. So this formula here, where Fi is the spline and Xi is the data in the fit. Now, important to emphasize here that the spline coefficients are now fixed, so those don't change anymore. We're just using this as a function rather than Gaussian, for example. So the free variables in this are the position of the spline, and X, Y, and Z, the height or intensity, and then there's also a constant background term. So here is an example of using a PSF, using splines to model PSF. So again, we have the astigmatic 2016 challenge astigmatic PSF. And as you can see, that's on the top, and the splines at the bottom over the full range. You can see that actually does a pretty good job of capturing this PSF. And this is probably the primary advantage of this approach, is it's actually very versatile. It doesn't have to look like a Gaussian. It can be basically anything. For example, this is double helix PSF, again for the challenge, and then at the top and the bottom is the model. And again, it makes a very good approximation of that PSF. Another advantage that it has is it has lower systematic errors. So here we've analyzed the challenge astigmatic PSF-B data. So this is examples of B data on the left. If you analyze this with Gaussian fitting, which I've done this using 3D Gaussian, because that's another thing that I worked on. And you plot, okay, what's the mean difference between where this thing told you the bead was and the actual bead was as a function of Z. And you see that at least in X and Y, the error is actually quite large. In the context, we're calling this wobble. And so there's a fair amount of systematic error. Can be a fair amount of systematic error when you use Gaussian fits. The spline doesn't really have that. It's basically, you basically took the spline from the beads. It doesn't give you very good, very little systematic error. And again, I emphasize that both algorithms use maximum estimation to fit the data. It can also be more precise. So if you have, in this case, I similarly did just using purely astigmatic PSF. This is an example on the left. In X and Y, you see the Gaussian spline are pretty similar. But in Z, it's actually precision is a little bit higher, especially at a higher Z. And I think this is in part because as you go to higher Z, PSF is less and less Gaussian shaped. And so yeah, the spline does better. Because it basically does a better job of matching the PSF, we can fit it more precisely. And finally, I said, okay, well, everybody uses Gaussian because they're fast, right? So is this, how much am I going to pay if I decide, okay, I use splines instead of Gaussians? Well, I'm going to reference it again to the Gaussian fitter that we have access to. That I have used is the 3D Gauss term. So that's the Gaussian, and then C-spline is the spline fitting. And I did some calculation, some testing of the runtime as a function of object density. You see low density spline is about 2x slower. In this case, the algorithms are a little bit limited both by just basically bandwidth, getting images off the disk, putting the results back on the disk, and so forth. As you get to higher densities, and the fitting part of the algorithm becomes a larger fraction of the overall time, and the spline starts to lag a little bit more. But at the densities here is only about 3 times slower. So with that, I hope I maybe convinced you that cubic splines are a promising approach, primarily because they're very versatile, but they can also have lower systematic errors, can be more precise, and they're not going to be like, they're not orders of magnitude slower than just using Gaussian infinity. If you're interested in looking at this a little bit yourself, we have the source code on our GitHub repository. And I'd like to thank Joan Lab and the Center for Brain Science and the Advanced Imaging Center at Harvard for their support. I don't know whether I'm supposed to take some questions or whether I'm supposed to just go to the next person. But I also have a poster.
We have time for one question. What about the computation time? So it seems that the C-spine is slower. So do you expect to have better results with GPU?
I'm not really sure. I don't have that much familiar with GPU programming, so I couldn't say. I think that it's primarily slower because it's more memory intensive than Gaussian. Gaussian you just have, so basically it's a calculation, you have a bunch of exponentials. The spline you have to remember all these coefficients to calculate it across the dimensions. I don't know whether that would be helped by GPU or not. I guess GPUs make everything faster. So.
Thanks again. So now I can call Silvia Calabrese, which comes from Italian Institute of Technology. She's now in internship at the EPFL. Okay, and she will speak about the machine learning in super resolution.
Good afternoon. Can you hear me? I hope so. So I'm here to present a study on the application of machine learning techniques on 2D and 3D single molecule localization microscopy. I don't need to introduce myself. Thanks, Daniel, to have done it. So, why the use of machine learning might even help? We start to think about it thanks to the 2013 challenge. And there was a paper in 2015, last year, from Daniel Sage, and there were currently 55 software registered on the website. So if I want to use one of those for my data, how to choose among them? For example, I could choose among the winner and of my acquisition setup. To give an example, in case of long sequence, the winner was at that time Thunderstorm. And it's a very nice software, very user-friendly. There are 7 possible image filters, and then you can decide among 3 possible spot detectors. Among the user are it's really free to choose whatever you want. And among also, there are other features here. Gaussian, integrated Gaussian, so no splines, but whatever. But if I'm a user, how can I decide the best one among them? I say that looking forward, I'm looking also to the other solutions. We saw that there are 2 general trends here. There are software that wants to lower the influence of noise. I would say noise to say background and all the other kind of noise. And I can have this result with smoothing, filtering, basically thresholding something. So getting rid of some information. Or I can try to find a faithful model of the noise itself. And this is I've done something. Okay. And this is this is important because whenever I have a faithful model of it, I can use the Kramer rollover bound with the MLE, and this was already studied in 2014. Now, is it possible to use in another ways? I mean, to use the background in other ways? Yes. I would say yes. Okay, I'm doing something wrong, so. Hmm? Okay. Uh, okay, so yes, is it possible? And in particular, we found a solution using the machine learning in the detection step. What I mean is that we found particularly interesting the use of support vector machine, which is uh, as I said, a machine learning technique that is based on the building of a hyperplane that divides two or more classes. In our case, it's just two. So I have class 1, in our case activation, and class 2, which is the background. As you can see from the figures there is a lot of variety, both in the background and in the activation. And it's just a minimization problem, so it's an optimization problem that usually can be solved with a hinge loss function, for example, to take into account non-really separable classes. And it is very simple. The only parameters that the algorithm tune are these W and B. Here is more clear, I think. I mean, I can take from my dataset a bunch of, in this case, are patches 7x7. Some of them I will label them as some of them as positive, the other one as negative. The positive are the one that has the activation. What I'm able to do is to have a model that can be used on test set. Test set is a machine learning way to say, but here I can just say my data set. So data that weren't seen before by the algorithm. And this is nothing else than a spatial filter, thanks to which I have that output. The output is a real number, confidence level, and X there is the instance from my dataset. Now you can say, okay, so even in this case we have a threshold. Yeah, but we have a threshold that is decided by the algorithm, so it's much more easier from the point of view for the user because it doesn't have to decide anything. And in particular, this is a very general approach. The use of SVM is just we decided to use the SVM, and in particular to use it just with the intensities. But there are many other options. This was particularly useful in case of the 2D example because the support vector machine can give X and Y that can be refined with a localization algorithm. Now, talking about a localization algorithm is kind of out of the scope in this talk, but just as I give you a reference, which was a paper of myself from this year. And we used logarithmic linearization to have the problem invertible. In this way, I mean, it's not really invertible with just one step, with just one step, just because we have noise. So there are, it's kind of iterative minimization of weighted least square error. Now, below there's a table of results. I hope it's clear, but I'm afraid it might not. And we compared the accuracy and Jaccard measurement of our method that is on the left with Thunderstorm, which was, as I said, the best method for a long sequence, and also the centroid. So in our case, the three tables refers to different data sets and that increase in terms of difficulties. As you can see, the accuracy, our accuracy is much better, and in terms of the Jaccard index, we can say that our results are comparable to that of the Thunderstorm without the need of, fine-tuning parameter or decide if, in my case, it's better, I don't know, to use wavelet rather than to use other kind of approach. And so to summarize, SVM can be seen as a threshold-free method. It includes the background in a spatial filter. I mean, I don't need other pre-processing step to get rid of the background, but I can include it. And the optimization decisions are done by the software developer, not by the user, which is most of the time unaware about what is the meaning of the parameters. But I must say that we are still working on the extension of real data set, so the results that I propose are on the previous challenge on synthetic data set, and also the 3D extension to it is still ongoing. Still, it's very, I would say it's very juicy, because learning is a paradigm, so changing descriptors, algorithm, and of course it will change also the resulting model depending on the problem. And that's all, so thank you for your attention.
Maybe I can wait out to switch the computer. So we have time for one question. Okay.
Is there a particular reason why you chose FBN? Did you look at other algorithms that people use? Was there a reason why you chose SBN? Or did you look at other algorithms like random forest to see how they performed?
So your question was why SVM and not random forest?
Yeah, or any other one.
The reason is twofold. The first one is that because of my background, so it's not a really scientific answer. I mean that I mostly in SVM and there are lots of implementations. There's a linear, for example, just and there are many other. I mean, it's way easier to implement. And it's also if you decide on forest, so you can be more about the transfer learning paradigm. So the idea is that to use the studies that were done in other fields in machine learning and especially machine learning applied in computer vision, use them to transfer it to this program. And in case of the transfer learning, it's just a way to change a little bit that weights that I showed before in the special field. I hope that I answered.
So it's a pleasure for me to have Ricardo here. So it's the famous author of the famous software which is QuickPad. But now there's a new software and we want to learn more.
Thank you. And thank you for organizers to bring this to give this talk and show you a little bit of what I'm doing. Or we're doing it well, sorry. So I want to tell you briefly about QuickPalm and then how we're transitioning to another channel. So I guess there's not much I can tell about QuickPalm because I mean it was published in 2010, and I guess most of you at some point either tried it or use it in your research. It's an easy algorithm in the sense that it's based on center of mass. It's extremely fast in terms of computation. And one of the things that I really wanted to do with QuickPalm was this bit. Too many letters. This bit here, where I wanted actually to massively parallelize the calculations. So pretty much what QuickPalm is trying to do is to run each frame in its separate thread, which means that I can actually do calculations quite fast. Once we started getting citations on QuickPalm, it was quite interesting because I started looking with who is citing it. And about 30% of it is reviews, 40% of it is about its biological papers. And the interesting part is 30% is other algorithms that are ranking against QuickPalm. And the reason why it was quite funny, the first paper that ranked against QuickPalm, I thought, damn it, now someone has something better, no one is going to cite me anymore. But they kept on citing, and everyone kept on comparing against QuickPalm. But this also highlights the fact that these challenges are quite interesting because they make a fair comparison between algorithms. And in a way, QuickPalm is going to be the best algorithm, but it's actually nice to have a third party doing those tests because many authors can be unfair on these comparisons. So what are we up to meanwhile? I started the lab, and with the start of lab, I started with the team developing NanoJ. Now, NanoJ is actually just a library for imaging, but it's a library that allows you to do OpenCL integration. So pretty much what we try to do with NanoJ is to convert your gaming workstation, which is not that expensive, into a cluster. So every time we start using OpenCL with our calculations, we end up with 10 to 100 times speed up in terms of calculations. So this enables us to do a lot of things in terms of image analysis, but most importantly, it enabled us to get to these guys that you see here. One is virus mapper, which is a single particle analysis algorithm that Dave is on the audience. He's going to talk about it on Tuesday. And the other one is SURF. So for the second half of my talk in 5 minutes, I'm going to tell you quickly what is SURF and why is NanoJ enabling SURF at the end of the day. So SURF came from this. You've all seen this before, you're probably fed up of it, but there's one important part here, which is this one. And if you start thinking about electron microscopy, it's been several decades since it gives us the same resolution that we get with super resolution. So you can argue about microspecificity, and you can see things that you couldn't see before with super resolution because we're using fluorescence. But one reviewer number 2, right? One reviewer on the grant that I wrote for Human Frontiers told me this would be a great project for cryo. And although I was really nice, it's arguable, it's true. So the question is, what can we do with super resolution that we can't do with EM? Well, the easy answer is lifestyle, right? And to do lifestyle, you really need to decrease the amount of laser power we have on your samples. So that means that we really need to go into high densities of proofs. And that's what SURF is about. It will work very well for low density, but it works amazingly well for high density. So I'm just going to briefly show you how it works. So here's 100 frames of a sample illuminated at 40 milliwatts per square centimeter. So what we do is we break each pixel into a virtual pixel. So we create subpixels within each pixel. And we borrow some ideas from radial symmetry. Radial symmetry was one of the best ranked algorithms on this context. And instead of using radial symmetry per se, we converted radial symmetry into a new mathematical transform. So that mathematical transform, what it does, it tries to map regions of space that are highly radially symmetric. What that means is, whenever you have a through fork that's highly radially symmetric, and if you apply this transform, it pretty much maps out what we think is the center of that through fork. So it ends up, right. And we do this for every subpixel we have here. Now, you might think, well, that's going to be massive in terms of computation. And that's the reason why we use peak localization, so that we compress down to wherever we need, peak detection, so that we can compress down where we're actually localizing those peaks. And that's where NanoJ comes in, because since I'm doing this on the GPU and not on the CPU, I can massively parallelize these calculations. So all this is done almost in parallel, simultaneously. So we do it extremely fast. Right, so how does this look like? Once we start applying that spatial transform, we end up with something like this. And pretty much you start seeing that, oh, okay, so there's some peaks that are being extracted here from these profiles. And the cool bit is you visually, by inspection, you can't even see particles playing there, but the algorithm is already starting to pull those guys apart. So I'm just going to speed this up a little bit. And here we go. So now I'm doing 100 frames per second. And immediately you start getting structure from there, and you start seeing that we start getting super resolution. However, what I'm showing you here is just increasing the speed at which I'm applying to the transform over 100 frames per second. Now, this is where we can actually borrow some ideas from Sophie, because this is just plotting this information. But if I start using temporal correlations on this data, what happens at the end is that I start clearing this out, and I end up with a pretty decent reconstruction of what those filaments are in space. Good. So, what have we done with this? So, bottom line, for a very dead cell and fixed, SURF is very similar to ComStor. And you might even use other algorithms. You'll get equivalent results, or sometimes I'm not even declaring that you're going to get better precision than what you get with SURF. But for a live cell, we would with GFP using milliwatts instead of kilowatts, which is typical with what we use on this one, for example. we end up with this. So what you're seeing here is just microschools labeled with GFP. And we often get better resolution than SIM at faster speed with less illumination and no special optics. So this is just pretty much your general turf or wide field microscope that we're using at the end of the day. So what are we using this for? We've been looking at mitochondria, here also, and into immunological synapses. This is data that we've acquired in cooperation with Gary in the audience, with Dylan. And the most interesting thing about this is that because SURF can deal with very large amounts of fluorophores overlapping, we can actually use almost any microscope to get super resolution image. So here we're using a classical wide field microscope. This is a laser scanning confocal microscope. And because SURF is purely analytical, you can really apply and extract super resolution out of these systems. Just to round it up, for example, here is a spinning disk data set. SURF is not 3D. What you're seeing there is just optical sectioning, and you're extracting super resolution in XY. But you can actually run this on a spinning disk, and the algorithm is so fast that it's just with 100 frames that you can actually get a very decent reconstruction. So this is a live QSL expressing image. And that's it. That's my 10 minutes. This is a small movie of everything being developed until now, or until last week. And anything people to thank, but the synapse stuff that you've seen here was in cooperation with Dylan, and Jason is one of our big collaborations with which we do various imaging. And thank you for listening.
Thank you for these impressive results. Is there any question? Just one question. Yes?
Are there any artifacts and what are the trials?
So artifacts, yes, they are. So SURF is relying on detecting oscillations to infer if fluorophores exist in space or not. So cell motility is the big problem. If you have a fluorophore that is moving during these 100 frames that we're using to calculate oscillations, we're not going to get any contrast out of it. Or we might end up generating more noise. And if you see some of our T-cell movies, especially ones where you have an actin flow that is huge, you will see that regions where the actin is moving highly, you almost get no signal out of it. So yes, cell movement speed is going to be one of the biggest problems. We're almost using the priors. Our prior is the radial symmetry. And the fact is, noise will generate some radial symmetry from time to time. So SURF will keep some degree of noise that exists already on raw images. Fortunately, noise is correlated in time. So the moment that we start running autocorrelations over time, we start clearing up that noise.
Just one other curious thing. So the fact that you have this radial symmetry, is it really critical to have a good PSF, which is radial symmetry?
To some degree, yes. We've actually been asked by reviewers to look into that, and we created glimpse of PSFs and shows that we still have conversions to the center of that PSF. Of course, if you go into the stigmatized PSF where you actually have two peaks, then the algorithm will map those two peaks there. So there's still some dependence on symmetry for PSF base.
Thank you very much.
Thank you. Alex
Herbert, thanks to coming also to So Alex is the author of PeakFit, which is also a very famous software. He participated to the challenge in 2013, and he contributed a lot to the organization to debug the system of this challenge. Thank you, Alex.
Thank you. So I'm just going to talk about what we've been doing at the Centre at the University of Sussex. So we started this in 2011, and we really wanted something that kind of ran cross-platform with minimal hardware, a simple install, it ran fast enough that you could, you didn't have to go away and come back after lunch to look at results. It kind of runs in seconds rather than hours. And we've been applying this to just 2D wide field images of yeast, and mainly looking at the analysis of diffusing molecules. So sort of not really interested in structure at all, but in counting, clustering, diffusion speed of complexes. So looking at what single molecules are doing, so binding DNA and non-binding, things like that. So I put together a whole bunch of ImageJ plugins. So ImageJ can be used to just load your images. It has loads of plugins anyway, and you can use our software with it too. They're fully supported with macro language, so you can record what you've done, play it back. You can build scripts to chain together different analysis. There's things to do spot identification, fitting, looking at the results, clustering and tracing. And then there's image simulation performance benchmarking, which I've got a poster on here. I can talk to you about that. So using the VG update mechanism, the installer software just installs with one click into VG. We've been using Google Analytics since March, and that's shown we've got 340 users around the world. And you can see that most people do their work not on the weekends. There's a number of sessions over time. So no one's doing super resolution on weekends. So the main plugin we've got for doing localization is a basic simple method where you split your image up into frames, you identify the spots, and you process them in rank order. I've put has neighbours, I'll talk about in a minute, but basically most of them have no neighbours. You fit them to a single spot. If it's okay, store the results, and if it's not, you have some sort of logic to decide whether you should continue or not. I mainly work on Palm with really low density stuff, so this works for us. What I've developed for this challenge is like a bit of an add-on that deals with high density data. So if you do have neighbours in your region, then you can fit the neighbours as well. If that's fine, then you save the results. If it's not, you go back to try and fit a single spot. And another concession is, for a single spot, you can try and fit them as two spots. If that's a better fit, you can store those results. So the sort of smoothing we have. I'm pressing. So you just do image smoothing, and then you find maximum. You see in this image, there's loads, of which some of them you pick out by eye. The red ones are the ones that ranked highest, and they go red to blue. So apologies, colourblind people, but most of the good spots that you can see are there. And then you fit a 2D Gaussian, least squares or maximum likelihood visits slower, for about 20 to 100 times slower, depending on how many neighbours you have to fit. So in this case, you see the first spot here is quite easy, just fit a little region, it all works. If you get to something like this, you have to fit the neighbour as well. Otherwise you get localizations inside the wrong place. When we're doing this, we just store the fit that we're interested in because the other one we fit later or has been fit already. And then we just filter spots based on the signal width, the drift they have, and precision. So if you take something like this, with all the candidates you end up with, these are the fit results from this image. And results can be presented in ImageJ tables or reconstructed, but this table would be interactive, so if you click on the results rows, they show on the image those localizations. We've got visualizations, such as reconstructing the full Gaussian PSF drawn, or a super resolution reconstruction of this data set, which you overlay the original image. You can see the sort of localizations of roughly the structure is. Talk a little bit about the challenge, because it's taught me quite a lot about the software we're developing. The low density data sets, I think that about 90% of them, and 84 for these two low density ones, were just fit as single spots. But it's in the high density data sets where this one has got 50% was fit purely as singles, the rest of them have to go through the additional workflows of being fit to multiple spot. And this one, 22% fit singles, and it's this data set where the method didn't work very well. I just looked at all the scores across each of the data sets and compared mine to everyone's using Z-scores, and it's this high density data set where the method kind of falls down. It's not really what it's designed for, so it's not unexpected. So if you want to know any more about this, you can go to our webpage. There's a user manual, contact details for me. I can answer any questions you've got. All the source codes on GitHub. There's the super resolution project, which is 8,000 lines of code, but there's a super project which you can check that out and checks out all the projects that I work on with ImageJ. And there's a lot of free, useful software there if you want to have a look. Thank you.
We have time for one question or two. So maybe you did not mention that PeakFit are also a very good website, very easy to use. If you have some comment on that.
Well, the idea is to try and stop people coming to Marcus and asking those questions. So try to find good software documentation so that people can use that first off to work out how the plugins run, what the parameters are, etc. And that's the key. The key to this is to try and make it usable. There's a lot of software out there to run command line. It's not all encapsulated. You can't see the images once you've got the parameters. So this is one package that puts it together and tries to make it.
Thank you. So now it's the turn of Jonas Ries. It comes from the EMBA and Heidelberg. So he has the author of the software SMAP. And I should also thank him because he participated in the challenge and he participated for all modalities.
Thank you. Yeah, while we wait for this screen, so I'm here at EML in Heidelberg, and my group is developing and applying advanced super resolution microscopy methods. And the software we are using in my lab, basically I wrote it, and it's called SMLMS, the Super Resolution Microscopy Analysis Platform. There, it's almost there. Yes. Great. So it's really great that we are meeting here to compare our software and that so many people participated. But I think it also kind of points into the direction of maybe a problem in the field. That is that so many groups are developing their own super resolution software from scratch instead of using existing really good software packages. This continuous reinvention of the wheel kind of, it wastes some resources which might be better used to implement new ideas, new filling algorithms, and so on. So the last time I rewrote our software, I had this in mind, and I tried to write it in a way that it's as modular as possible, also as easily extendable as possible. Yeah, for our biological collaborators mainly, and also now for us, I try to have an easy to use and customizable user interface, and of course I want to have all the great functionality around in the internet and in the community. So here you see an overview about the different analysis steps of typical super resolution software. You go from camera images to the list of localizations, which then you can render as super resolution images, and then you have to analyze those, or you directly analyze the localizations. And all these steps, of course, depend on many parameters which are usually provided by the user. So in SMLMS, in our software, all these steps are encoded in what we call modules or plugins. These modules are actually quite easy to write. You take a template, put in a few lines where you define the user parameters, which are then rendered in the user interface later, and then you implement your specific functionality without much worrying about the rest of the software, and finally copy it into the plugin directory. Although most modules are really independent from each other, they can talk to each other using a global parameter structure. This we use, for instance, to synchronize fields of the user interface. Also, every module saves its parameters, so afterwards you can always know what you did to your super resolution data, which analysis steps you performed. So modules can then be connected into what I call workflows. It's just really a list of connected modules which run on the dataset one after the other. And we use those workflows especially for the fitting part. This is a typical fitting workflow we use. And the reason why we split up the whole algorithms in so many individual plugins is because that makes it very easy to exchange some part of the functionality without worrying about the rest. So if one of you comes up with a better way of, let's say, doing background subtraction before the peak finding, you implement it as a plugin, and then you just exchange the module in the workflow and don't have to implement a user interface or a reader for the camera frames. So at some point, if you have so many plugins and want to define the user parameters, it gets a mess. You have to go through this list of maybe 20 plugins. You don't want to do this. So that's why we map the user controls, the input, onto one page in the overall user interface. So it's easier to configure. And these workflows can be easily assembled just with a mouse click. In my group, we mostly use the GPU-based maximum likelihood fitter from the Keith Lipke group. But in the software also implemented, or actually I just took the random symmetry in 2D and 3D, and the high-density Falcon fitter. 3D, we usually use, or we usually do with astigmatism, but in the software you can also use biplane or double helix. Something which we really like to do, and which is quite nice, is that we can fit already during the acquisition, also render and analyze our data during the fitting. And with this real online analysis, we can immediately see if the data set is good, or if we want to go to the next region of interest or the next sample. For the rendering, I want to have some kind of Google Map-like feeling to explore the data, to move around, to min, to bound. So I implemented a fast renderer. Again, there are different plugins, different functionalities, how to render the data, and you can write your own. But we most use a Gaussian renderer, where every localization is rendered as a Gaussian, with a width proportional to the localization precision. Not the same, but proportional. Then we have different layers, and each of these layers can be rendered individually, and in the end, the layers are just overlaid on top of each other. And this allows us to display different data sets, different channels, or just, as done here, overlay a diffraction limited image with the super resolution data. And we can basically filter any property of the localizations. Because it's really quite easy to write plugins, we already have quite a few. And at some point it gets messy, it gets difficult to find the functionality you're using most, or you want to use. And this is why we have a configurable user interface, where you put the plugins you use on a daily basis at well-defined positions, and all the others which you usually don't use, you can still access from the menu. Also, it's possible to define optional parameters together with default values, which can then be hidden in the user interface, and this makes it easier to use the software, especially for our biologists. I briefly want to show you two plugins we use a lot. One is a real-time 3D viewer, of 3D renderer with several render options. It can do these stereoscopic images with the cyan red glasses, or with these Google Cardboard-like viewers for your cell phone. It works, but honestly, for me, it gives me mostly a headache. Another quite nice plugin which we really use a lot is the so-called ROI manager. It can automatically or manually segment your data set into regions of interest. You can annotate them, also automatically or manually sort them, filter them, and then pass all these regions through an analysis pipeline. And finally, do a global analysis on all your data. And if you come to my talk tomorrow, then actually most of what I'm showing there was analyzed using this ROI manager. So to summarize, our software is quite modular and I think quite easily extendable. It has a hopefully easy and at least customizable user interface and a lot of functionality. At some point, if I find time, I might publish it as open source. Until then, just write me an email if you want to use it. So it's based on MATLAB. On one hand, this has an advantage because many people know how to program MATLAB, and maybe are more likely to provide own code for this. On the other hand, of course, not all groups have MATLAB, and then one has to use the compiled version. Then not everything is MATLAB. Some parts are written in CUDA, C++, and Java. So in principle, we can also use plugins written in other languages and other platforms, and then we have to interface to them. And we interface actually quite closely with Micromanager and ImageJ. So now I think I still have a minute, and since this is a 3D competition and we spoke a lot of simulated data, I want to show some real data, which we image just using the standard astigmatism approach and DNA paint, just to show that in practice at least our software can do 3D fitting and drift correction. So these are of course microtubules, and if we now use our 3D viewer to construct a side view like this, you see that all the microtubules appear as this hollow cylinders. This is clear, the fluid force cannot enter the microtubules themselves, so in the center you always have this black line, but you need a really good resolution to really see this. But if you have this resolution, then you can really think of using super resolution microscopy to segment microtubules and cells, even at these really dense regions. You just have to follow these black lines through the cell. So in the end, I want to thank all the people who struggled with all the bugs and with the daily updates always, and gave me still constructive feedback, and you for listening.
Is there any question? We have time for 1 or 2 questions. Yes?
Yes. So how would the support of other algorithms work? Would developers have to adapt their interfaces into your MATLAB codes, or how do you see it happening?
So I would imagine that you have your algorithms there, and then at some point you read the documentation once it's there, and you know how to interface it, because it's not so many different data formats there you can pass on. I think for the fitting, you either go from camera images to list of localizations, or you do something with localizations and so on. So it's quite straightforward to understand how to interface it. And that is probably just a small wrapper, a few lines to put the right data into your algorithm and take the right data out of it again and pass it to the next module.
Thank you. Other question? And maybe I have one. You said something very interesting for me. You said that you have a way to find an optimal way to find the parameters somewhere. Could you comment on it, because that's a critical point?
Okay. No, I think this I didn't intend to say. No, in the end, I said that. So with me, it's mostly user-defined parameters. Of course, if we can avoid them, we should. But we have optional parameters where you say usually this parameter works, so you don't even show it, and the biologist doesn't play around with it. But sometimes you still want to tune this parameter yourself, and then you show it. And, yeah, optimize it.
Thank you. Thank you, guys. So now it's time to Guy Hagen. So Guy Hagen is the co-author of Thunderstorm. It's a very well-known now software who won the previous challenge. Guy Hagen recently moved to Colorado University. Thank you.
All right, thank you very much. It's a pleasure to be here today to tell you a little bit about Thunderstorm. And as he says, I was formerly in Prague, where we did this work, and now I'm at the University of Colorado in Colorado Springs. So as we know, there are several steps involved in analyzing single molecule data, and Thunderstorm offers a variety of methods for each of these steps. The methods can be used in any combination, or nearly any combination, and each of these different combinations represents a different algorithm. And so some of the steps are shown in this flowchart. We start with image filtering and feature enhancement, and then finding the approximate positions of molecules, which we call detection, and then the subpixel localization and post-processing, like drift correction and so on, and then finally rendering. And these are shown here using some example data. So here's on the left is some raw data, and then there's 2 examples of the different filters. There's a denoising filter, which is just a Gaussian filter in this case, or this bandpass filter based on wavelet transformation, which we call a feature enhancement kind of a filter. And basically what this is set up to do is to identify features in the image that are the right size to the single molecules. And then using that to find where the molecules are, we then apply a threshold, and then finally on the right-hand side you can see the detected molecules. And this is what the program looks like. It's an open source program for ImageJ. Okay, and we can do 2D and 3D analysis with biplane or astigmatism methods at the moment. And we can also create and evaluate simulations. And we have really extensive documentation. There's a website, including a wiki. There's a forum. The software is still under development. It automatically updates in ImageJ, but you still have to click it. It's not the super Fiji time. And then each of the steps that I mentioned are in these kind of sections of the program, so the image filtering and subpixel localization and so on. And it was published in 2014 in Bioinformatics. We also have help online within the software. If you click on these little blue question marks, it pops up the relevant section of the user manual or supplementary information. On the right-hand side is a page from the website and the wiki, and all of those links along there are supplementary data or little tutorials or so on that help you use the software. And then as Sylvia mentioned, we have several different options for each step. So there's 7 different filters, or you can also choose to use no filter. 3 different methods for the detection, different PSF models. There's 2 non-fitting methods, or fitting methods with least squares or weighted least squares, or maximum likelihood methods. We have Keith Lidkey's multiple fitter or multi-emitter fitting analysis that can use either MLE or least squares. And we have statistical tests to determine the proper number of molecules in that case. And then finally, 4 different rendering methods. So then as Sylvia mentioned, well, out of all of this stuff, how do I know what's best for me? And so we've made a default button with a certain selection of this stuff that is based on a lot of testing that we did with simulated data and real data, including the challenge data. And this is just a recommendation that people can use. But more broadly, you know, as you get going in this, then you can start to play around with all these different kinds of settings. Also, the threshold can be just a value, but it can also be based on an expression. And this, we have found, works really reliably. And so that's part of our default settings. And so basically, our approach in the challenge, in the first round of the challenge, was to take the training data sets and run the training data with all the different combinations, like 75 different combinations, and then to find the best one in terms of the F1 score or so on. And then we used that same group of combination settings for the challenge data. So this is the results table. The data can be imported or exported in many different formats. We have interactive histograms of each parameter. So what's shown on the right here, once you have the results that include the number of molecules and which frame they were in and the number of detected photons and so on, you can then plot a histogram of that. And then in the histogram, you can draw a box and then click that apply Roy to filter. And so then it automatically chooses those, that range of values and removes all the molecules outside of that range automatically. And then once you have the results, there are several post-processing methods. There's a density filter to remove molecules that are really far away from any other molecules. Like if you have a background spot over here someplace, these can be removed automatically. There's 2 different methods for drift correction, either the correlation method or using fiducial markers. Both of these also work automatically, so it automatically detects fiducial markers as spots that don't blink and does the correction based on that. And then just briefly something about the simulations. We have tools for creation and evaluation of simulations, and that's shown here. And we can use a density mask and also a background mask. And an example of the density mask is shown on the right. The Siemens star has a different brightness for each spoke, and then each of these brightnesses are interpreted as a different density. And so then you can see that in the result, each of these spokes has a different density of molecules. And then the simulation engine produces a stack of images with the blinking molecules in them, and then we have the ground truth for that, which is one table, and then after analyzing the data with Thunderstorm, you get the results table, and then you can see the matching between these. So the green ones are the ones that match, and the red ones didn't match. And then it automatically pops up a table with the precision and recall and the F1 score and this kind of stuff. Another thing that I think is really interesting is the idea about creating realistic simulations that are based on measured parameters. So everything in these kind of experiments can be measured. So the number of detected photons, the background, the full width at half max for the molecules, the camera parameters, and all of that stuff can be measured and then used to initialize the simulation. And in this case, the density mask that we used was a wide field image. And then we have the real palm data and the simulated data. And then this allows us to ask a question like, well, how close is the real data to the simulated data? And this, I think, is a really interesting application. And then there's just an example here of some of the different things that we've been working on. On the top is it's an epithelial cell called an A431 cell. And it's expressing the growth factor receptor ERB3 labeled with the YFP citrine. So we have the wide field image and then the single molecule image. And then once you have the list of coordinates, you can then calculate the density of these and make a density map and then overlay that on the wide field image. And this also, I think, is a really neat application because it's information that you can't really get another way. That feature is not in Thunderstorm yet. And then on the bottom, we have a storm kind of a method where we're doing a two-color labeling of the cell nucleus to look at two different activities within the nucleus. In this case, replication of DNA, which we did using EDU, Alexa 647 EDU, and then transcription of newly synthesized RNA molecules labeled with the fluorouridine row. And then finally, our other project is SIM Toolbox, and this is for structural illumination microscopy, and it has a similar feel. It's Thunderstorm in that you have several choices for all the different steps in the algorithm. And we have several different options for optical sectioning SIM, super resolution SIM, also SIM using Bayesian estimation, and this was published in bioinformatics as well. And on the bottom here, these are live cell movies of it's a lysosome-associated membrane protein labeled with GFP. And so then finally, I'd just like to thank the Thunderstorm team. Pavel Kuzak developed all of the initial stuff in MATLAB, and then Martin Ovesny is the real author of Thunderstorm, and he continues to develop it to this day. And also, Tomasz Lukasz is here now at EPFL and was part of the team working on structured illumination. So thank you very much.
Thank you. So, question in the audience? No question? Yes.
Okay. Since you're implementing so many algorithms on Thunderstorm, I guess that would also make Thunderstorm one of the best platforms to rank these algorithms against themselves. Are you doing that? Is that a feature that you're thought about?
Yeah, so the question was if we can use Thunderstorm to rank all of these different possible combinations of filters and detectors and so on. You can do that. We haven't really, we've done it a little bit, and you can find in the wiki, in the web page, a little bit of that kind of analysis. But you can use the macro feature of ImageJ to set up something where you would systematically go through a bunch of these parameters, and then you can compare them based on the F1 score or another kind of a metric.
And you have, okay, now you have astigmatism biplane.
Yes.
What's about the double helix?
This we didn't get to yet, basically. And the reason for that was that we didn't want to put in anything that we hadn't tested extensively, and we didn't have our own data for double helix, and so we didn't do it. Thank you.
No, it's not true. It's RV. No, it's RV.
I'm sorry, that's my bad.
So now it's RV old, so it's coming from January of France. So he has this software called VREX, and VREX already participated to the challenge in 2013. He won also, he had a very good rank in the high density.
All right. Do you hear me? I guess. Yeah, so first I'd like to thank the organizer for putting together this very nice challenge, or challenging thing, I should say. And also for giving me the opportunity to present VREX here. VREX is a software I developed with the primary goal of reconstructing dense localization microscopy dataset. It's essentially a Bayesian method, and so I'll try to explain to you first the internal logic of VREX. Well, so you are all familiar with the localization microscopy, of course. So when you have an isolated spot like this, everything is fine. It's usually quite easy to fit. But when the spots start to overlap, the challenge to reconstruct an original image of fluorophore position is actually much more challenging. And still, it's a very interesting problem because it allows you to be more accurate, even when it's quite sparse. It allows faster acquisition and also higher time resolution if you wish to do some live imaging, for instance. So, as I told you, it's a Bayesian method, and so to do that, you have to somehow abandon the idea of localization coordinates and work with images of fluorophores, where the pixels here are actually very small, and you have an image you want to reconstruct IS, and measure the image. And actually, once you know the original image at very high resolution, it's usually quite easy to find the probability of a measured image because you know quite well your optical system, the noise in the system, etc. So this operation is easy. The reverse operation is much more challenging, but to help us, we have the bias rule, and we can actually invert this. So find the probability of IS knowing IP. And it's actually the inverse probability, so the likely P of IP knowing IS, times some prior, which give you some knowledge of the original image without any measurement, and a normalization factor. The prior is very important here because it tells you that still you won't have any kind of image originally, right? You will have images that will be very sparse, and that's, of course, what is very important. And so let's first try to define this prior of IS. So it gives you the probability in one position to have a fluorophore. So if you have 0 fluorophore, so it's the majority of the probability you have 0 photon count at this position. And now if you have a fluorophore, well, you have a distribution of number of photons emitted at that spot. So that's how it looks, and all the pixels in IS are independent, so that basically you just multiply all the P of IS to get the whole product. And now you have this posterior. So if you want to detail now the likelihood, well, what we take in VREX is simply a Gaussian distribution telling you that the original image goes through the PSF, which in that framework can be absolutely arbitrary, and gives some measured image with some noise, where the amplitude can also be fixed. So you see that in this framework, it's very flexible. You can adjust the noise level as you want, and also the PSF can be totally arbitrarily chosen. So we've tried with astigmatic, but also thanks to the challenge with double helix, and the result seems to be somehow reasonable. We have also tried biplane, but also multiplane. So when you have a whole stack, and this is, for instance, useful for a single molecule fish. And so how it works, that's where it becomes a bit more technical. So to simplify, let's say we have 1D image instead of 2D. So we have measured pixels here, which are somehow the constraint of the inference, C1, C2, C3. And what we want to reconstruct, okay? So you have many more pixels. And basically, okay, if you have some photons here of a fluorophore, you will have a response on several measured pixels here, okay? And in the very same way, if you have some photons here, it can come from several super-resolved pixels here. And when you do the inference, actually, you have the eye of the eye, if you like, all influence each other, and that's where it becomes very complicated. And the idea behind BREX is to use the better approximation, so that's why it's a big reconstruction. And is some approximation which is usual in some domain of computer science, or also in the physics of spin glasses, where you neglect some of all these loops. So if you want to update the intensity on a pixel that you want to reconstruct I3, for instance, you will consider all the influences with the neighbors, but then you will neglect the influences on the neighbors themselves, so that you have something that is locally a tree. And so this approximation is very powerful. What I discovered is actually it doesn't give you everything, and converging algorithmically toward this approximation is actually very difficult. And that's where the novelty of BREX is really reside. It's really buried in the internal of this software. But anyway, that's how it works. And as I told you, it's iterative, so you will basically build some clouds of probabilities that evolve with time, so you iterate. And you see that even when it's very dense, it allows to find several positions that refine with time. And from this cloud, you can try to find some localization. Okay, so that's how it works. As Daniel was saying, it's working reasonably well. Notice that contrary to some other software, when you have some crossing, like here, you don't have that many localization in between here, just because in the algorithm itself, it takes into account the interaction of all the reconstructed pixels and avoid this kind of blurring in between. So that's quite interesting. And also, sometimes when you have this kind of data set, sometimes I guess it's hard to see here because it's too powerful. You have small localization here in the reconstructed image. But sometimes when it becomes too difficult, you just have clouds. And I think it's better to just sum these clouds and actually try to find some localization where you don't actually know where they are. And when you sum these clouds, you actually obtain reconstructions that are usually more reasonable than noisy localizations. Okay, and so we've tried this software also on different metrics. And you see that in region where it's extremely dense, like here, you have some error, but still it managed to find quite reasonable clouds. And an interest also is that we observe that the total intensity that it reconstructs is very faithful. So if you want to find how many spots you have, I mean how many photons are emitted, so if you want to filter the noise, usually it's extremely good. Okay, so now I will stop here. So I will thank my collaborators. So in particular, Timothy Lyonnais at the Transcription Imaging Consortium in Geneva, that has been a very big driving force. Also, one problem with the last challenge is that clearly the user friendliness wasn't that high. So I tried to remedy that. So now you can compile it for several platforms. You also have a Fiji plugin, and all the code is released publicly, open source on GitHub.com actually. And yeah, I'll stop here and thank you for.
Thanks, Hervé. So, question in the room. So we have time for one question. Yes.
Yeah, as always with these Bayesian approaches, I wonder how long does it take? And if it takes long, can you speed it up on the GPU like Ricardo does?
Yes, so what is very nice with the better approximation is that it's very parallelizable. So what I do to speed it up is to use SIMD of the CPUs, but it's not GPUs. But still it performs quite well. And also, yeah, it's very different from compressed sensing, where you have algorithms that scale very badly with the size and so on. Here it's iterative and it scales very well. So basically it scales linearly with the number of pixels you reconstruct and this kind of thing. So, I mean, it's not as fast, of course, as PeakFitting, but it's reasonably fast. And also when it's very, when it's sparser, it's extremely fast, but even when it's quite dense.
We have maybe time for the question. Okay, and maybe there is a microphone.
I can also scream.
I'm sorry, I focused on the back.
It's a simple question, because once you go into the point clouds, it's actually some degree similar to CERN. So that brings me to the question of, what about the temporal component? Did you think about starting also using the Bayesian priors into the temporal analysis, or how?
To do what? Sorry?
What about the temporal component?
Oh yeah, that's a very nice question that you already asked me. But no, actually, it scales badly when you have a very long data set, right? Because you would have to put everything in memory to do the right inference. So what you can do is to chop it in chunks of, let's say, 10 frames or so on, the relative time scale of your storm dies, let's say. That is perfectly doable, and we have to discuss that at the discussion later. But what I can say is that the better approximation and this kind of thing are very good at this kind of thing. I will stop here, but yeah, it would be a nice way to do it.
Thank you. So now is Anne Wheeler. She's coming from Hindenburg. So she will speak about benchmarking super resolution, and specifically about. I have already listened one time your talk recently in LME, so it was very interesting. So I would like to have a second time.
Thank you very much. Thank you to the organisers for inviting me. I have changed the talk a little bit, so many things that are me. I have changed it, so it's not quite so boring. So, what I'm here to do is talk about benchmarking super resolution, and the reason that I'm here to do it is because I work at the Institute of Genetics and Molecular Medicine, and it's a molecular cell biology institute. I'm also part of a super resolution consortium in Scotland, which is called the Edinburgh Super Resolution Consortium. So, at the IGMM, we provide biology, and at Heriot Watts, they have an expertise in engineering, so they provide that. And then what we did was we sort of got together a few years ago, applied for a big platform grant, which we were granted kindly from the Mathematical Research Council. So we basically got together and purchased several different super resolution systems which are open access. So we have people from all over Scotland and various other places come to use the equipment. We also have a PhD programme, and we run the only super resolution summer school in the UK, where we get the people who developed and pioneered the techniques to come and teach people from all over the world. We had someone from New Zealand this year, so quite a long so one of the things that I quite often come across in my capacity as a person who sort of has to try and interpret what's happening in super resolution to the molecular cell biologists in my institute is that they have a bit of a challenge in whether seeing is believing or not. So as we have seen several times in very good talks today, basically the way that SIM and single molecule methods work is by using image processing to improve the resolution. So you have a series of images which you then segment somehow, fit somehow, judge somehow, and reconstruct somehow. Obviously, the question is, what is that somehow? And if you're coming from a molecular cell biology point of view, this may be a little opaque to you. So it's well known that the effectiveness of the reconstruction is as good as the parameters used. And something that I see, because I look after many advanced imaging technologies as well as super resolution, is that journals for many other technologies are very specific in asking for the procedures used, and they want to know how that's been done and what the clarification of that is. So for FRET, and for high content screening, you have to be very specific, and there are criteria specified in journals and defined by people. Harvard recently did this for high content screening. But for single molecule methods, this is sort of more opaque, I think, as methodologies develop and we have challenges such as we do. So I performed a brief benchmarking sort of single molecule experiment, and it was done with my system, which sort of could actually do a little bit better than it does do. We bought a better filter there. So the images have background, and it's kind of high noise and low signal. So I ran it through various different algorithms, because this is the sort of image that people in my facility are kind of generating. So you can see that basically you run it through 4 different algorithms, you get 4 different answers, and actually because it's sort of so powerful and it can choose different things, in certain respects I guess Guy leaves it open for people to choose, but as Ricardo's question was, how do you compare it against one another to see what you get? Well, this is what people in my institute are doing, and then of course they're very confused by the answer because you've got 3 different answers there. So the question is sort of, how good is it? Because the data is the same, but the answer is different. How much information do we need to say that a single molecule localization image is indeed accurate, is indeed representative and correct for the raw data? Because obviously biologists worth their salt will want to commit themselves to something which they can be laterally proved to just be a mathematical artifact or something else. So these are 2 separate data sets taken from very, very different papers, both of which accepted for publication. This one has got, the raw data and a binarization of the image, which is interesting. And then they sort of put on a colour lookup table, which indicates where the molecules were. And they've got some information at the bottom about how many molecules there and what the area is there, which is presumably maybe measured by thresholding. This paper, on the other hand, had a supplementary data section which was 10 pages long and went into specific and extreme detail describing how statistics were done and which algorithms were used. But both of these got past peer reviewers, which is quite interesting because they're very, very different in what they report, but it's the same technique. So this is confusing for biologists because they don't know what they're supposed to be saying. So we use manufacturers' software, and the manufacturers, I feel like, say they're in a bind because they report localization accuracy. Although for this particular experiment, I used a primary and fab fragment secondary. They claim the localization accuracy is 7 nanometers. I think it can't possibly be, because actually if you use primaries and secondaries, you can never get down to that sort of minimal localization. So there's a problem there because it's not even reporting the right thing. What they export is a list of molecules and the X, Y, Z positions of that and no further information. Other manufacturers are doing a little bit better, but the question is, so this doesn't really help our biologists maybe because they still want to know, are my parameters useful? So something like branchless data can be really helpful here, because what you do is you can have your raw data and then artificial data which is generated, which should be vaguely representative of the raw data. I don't know that this is really the greatest representation of that, but that's what it kicked out. And what you can do is then you can start looking at metrics like the JAPAD index, so you can see false positives here, which are represented in the artificial and the raw data, and also true positives. And so you can, and also you've got some sort of false positives here as well. So you actually have some idea about how good the parameters you can put into the algorithms are, because you have so many choices about filtering and fitting. It's good to see whether they're good. But there's still some confusion about which specifically are the right parameters to report, which ends up faces like this in the facility, really. So there's a need for benchmarks, and I'd like to thank the organisers, and particularly the people who organise the challenge, for actually organising this, because it's so useful to the community. Most end users who are biologists don't have a computational background, so they find some of the maths quite complicated. I think it's great that we benchmark, because it improves the reliability and the reproducibility of the storm data, because reproduction is really what you do want to see. And I think for the biological community, exercises like this 3D challenge improve the trust in the technology. What it would be nice to see is some easy-to-use tools to quantify the robustness of the data. The SIM community did this, actually, quite recently. So SimCheck was published last year by a group coming from Micron in Oxford. And I think that these sort of things do help people judge that they've made the right decision. The other question I've come with is also sort of a useful ontology for single molecule localization. Because if we have something like this, it allows us to incorporate our resources very easily into other community resources, such as the open microscopy environment, which can be used to then document and kind of categorize the data and share it. So I guess, some thoughts we have had in discussions are around the XYZ positions, some concept of the uncertainty or the goodness of fitting. Which algorithm used would be very helpful, because there are at least 55 out there. And, do we need to commit ourselves to FRC, or is there anything else? And with those series of questions, I've provided no answers. I will leave you. Thank you for your attention. I'd like to thank Eurobioimaging for funding me in the first place to start this project, and also my other funders, EPSRC, BPSRC, and MRC. And, just to hand over to the next person.
Since we have a question, yes? Yeah, question. Do you have microphone? Okay.
If not, I can just repeat it.
But more of a comment, I guess, is that I think one problem that I ran into working with biologists is one of managing expectations, because they want to know, are there 1 million molecules or 1 million and one? And that's just not something that we can do. And on the other hand, they generate data that's just bad, and they expect something great out of it, and they say why.
Yeah, so I guess it's sort of the case question, you just comment about managing biologists' expectations, and also, sort of the quality of data. I think that that's where people in my position, because I am a cell biologist by background and I was trained in advanced microscopy techniques, may come in. Because, I think maybe, even having some sort of module at the front end of the software that says this data is too rubbish to even process might help people, because, or even on the fly, because I guess some people have fitted on the fly, and I know that Thunderstorm can do some processing on the fly, that, it's sort of after, 1,000 frames, it actually says you need to stop because your stuff's not blinking, or the only algorithms that is suitable for now is Sophie or, QuickPalm or something else, so people know, where they can go with their data. I would agree, I think that is quite useful.
Thank you. We have maybe time for the questions. We are not so, no? Okay, so that's, thank you very much, Tom.
Okay.
So now we can enter more deeply in the challenge, and Seamus Holden will present the data set, and specifically we speak about the 3D data sets of the challenge, the simulated ones. Thank you.
Okay, thanks, Daniel. And before I start, actually, I'd like to thank all of the competitors who put so much work into the challenge. And it's actually really nice to see everyone together here and meet a bunch of people who I only internet knew, right? And it's been really nice to see the challenge come together from something that originally we were like, is this ever going to work? So, and then hopefully it turned out quite well. So we're going inside the computer. And this is basically like the geeky talk about how the simulations worked. And then so I have my second talk at the end, which is kind of the general conclusions. But this is just the nitty-gritty. But just to give you the overview of why the heck we did this software challenge, right? And why we did a second one. I mean, I was competitive first, I didn't organize the first one. And why I pestered Daniel. This is how it went down. I got really fed up with my 3D data, 3D localization software. I was like, I don't know how good this is. I don't know whether my software is reading my data. I don't know whether I'm reading my data. And hence this challenge to the board. But before that even, there's just this point of does the software matter? And I think particularly in our field, unarguably, the move of super resolution to live cell has been absolutely driven by localization microscopy, high density localization microscopy algorithms. So it's one of those fields where software matters. And so challenges like this, the point is that it reveals the state of the field, it tells you where to go next, it tells users what to do. And so for the first time we could do that for 3D. And then the nice thing about doing a second challenge on 2D, it could tell us where the field has progressed. Okay, but this talk is about how the actual simulations work into that. So the key goals for actually building the simulator for the second challenge were to make the simulator as realistic as possible. And one of the biggest challenges for this was extending it to 3D. But then we wanted to kind of big up the 2D challenge as well and make it even nicer. But the biggest challenge was taking really quite complex point spread functions, which represent the main single objective super resolution modalities, and to build simulations around them. And then for 2D, we added in photophysics, and actually something that worked quite well was really experimentally derived noise models, which actually touches on something that the guy mentioned as well, where your simulations are relevant, right? And then realistic structures. And so actually, to be honest, the thing that took us by far the longest and was surprisingly difficult, and the whole committee was like arguing about how we're going to represent these complex point spread functions so this is the whole thing of like real point spread functions have curves, right? That's a theoretical PSF on the left, Gibson-Lang PSF, and on the right is a real experimental point spread function. This is no 3D localization aberrations. This is just your 2D PSF. And you can see not only it has lateral aberrations in addition to a very large degree of difference and variation in Z. So theory doesn't cut it when you're inputting it into a simulation. And this is, because you've got imperfect optics and refractive index mismatch and all this sort of thing. And once 3D localization microscopy works by adding further aberrations on top of that. And so you can see that the PSFs are really quite complex. They're not well approximated by the X-rays. And so you start with this question of actually how do we simulate this with any degree of realism? And our solution was to actually take experimentally derived point spread functions and use them as the ground truth for a simulation. So essentially all of the model point spread functions in this competition were derived from as high resolution, as high signal to noise as possible, sub-diffraction limited fluorescent beats. And then you run them through interpolation, co-alignment, normalization, and averaging to give you a 10 nanometer voxel size final super high resolution, super high signal to noise point spread function, which then goes into the ground truth for the whole simulation. And in the end, that worked quite nice. And what's nice about it is it's generalizable, so you can then use any modality. Someone invents it, like W. Murder's doing some lovely work on very unusually shaped PSS, which we'll be more into, and anything like that can be chucked into this simulator. Now, something that I think confused the heck out of a lot of competitors, and I just want to explain what on earth we were going on about, is this correction, post-hoc correction, which we applied called wobble correction. And so this is the idea that for a real PSF, the centroid, if you fit it with a Gaussian or many algorithms, will shift as a function of Z. It will shift laterally because it's an imperfect point spread function. So here this is a plot of the shift for rapid storm in X and Y as a function of Z. So you see you've got systematic offsets, so say half X distance, whatever, but which is kind of trivial. But actually you have significant and non-linear changes in the position of your apparently observed molecule. And if you want to compare meaningfully to a ground truth, you have to fix this. So this is why we made everyone go and localize these beads data, and we were so on that. And I'm sorry, but now it works. And this is just to explain visually. So this is without correction. So you see you have also, in the red curve here, you have non-linear shift compared to ground truth. And then once you correct for this measured shift, then it's all fine. Although it's really nice if software does this in-house, like say, for example, Hazen C-Spline just doesn't need to do any of this because it accounts for it. And that's obviously nice for the user and so forth. But that's the game. And so we had our 3D PSFs, and that all works. That was the hardest bit. And then we thought, okay, well, we'll make it as experimentally realistic as possible on the sample side as well. So we have blinky molecules with palm-like, experimentally realistic focus switching kinetics. We have experimentally derived signal and noise levels. So I looked through a bunch of experimental data that I had, and we tried to make noise levels that represent the major modalities of localization across a piece of fixed cell storm, live cell. And it's not signal to noise, right? It's signal and noise. And so live cell, fixed cell storm has very high photon count and a reasonable amount of background. Live cell PAM, although the photon counts are really low, usually you have very low background as well. And then you've got this exciting intermediate case of live cell organelles, where you've got kind of high, moderate photon count, high background, all sorts. And so we could do this and simulate realistic data. We also extended the structure. So for better or worse, right, we're kind of stuck with microtubules as the test structure. And they're great, but when they're prone to certain classes of artifacts because you're analyzing one-dimensional structure. It'll come back, right? So it was really nice to simulate an extended structure, which also kind of is supposed to be a little bit like the endoplasmic rectangle in a very approximate sense. And this, in principle, avoids these sort of artifacts and gives you sort of a realism in your structures. And then we do nice, accurate noise models. And that's your simulator. So you start with a continuous structure, populate with fluorophores, add photophysics, check it through your point spread function simulator, add EMCCD and shot noise, and then you've got your final data set and competition, which Daniel is about to talk about. So just to review, so we have a nice simulator that is in principle as realistic as we could make it. All the code is publicly available on GitHub. That reminds me, by the way, competitors who talked, the internet has already told me that not all of your GitHubs are working, apparently. So check it out. All the data sets will be freely available. It's going to be a currently running challenge. So we'd like to turn this into so at the moment it's like hack together, we can run the simulator, right? But it'd be lovely to turn it into a generalized simulator that is usable. And that's definitely on the cards. And maybe spread functions. I think SCMOS is an important and growing challenge, and it wasn't addressed in this competition yet. It's in the future. And finally, I would thank all of the committee, particularly Daniel Sage, who has done the field a huge service by starting this whole software challenge. And the committee worked very hard generating the experimental PSFs, organizing, arguing, all of the contributors, so all of the competitors, but there's certain contributors who either kicked their ass in the initial simulations in the interest of various things, or just worked really hard to make the competition better. And the organisers of the conference, and thank you very much for hosting us. It's lovely, and members of my conference group involved, and yes, all the competitors again, and our sponsors. And I'm happy to take a question.
Any question? Maybe a microphone.
It's very simple. What's going to be the next contest?
Oh man. Okay, so what's going to be the next contest? You know what? You can answer that because you're on the round table, which I'm moderating, and that's one of the questions, right? I mean, I have my own opinions, which maybe I'll chip in with, but look, SCMOS is really important, right? Ultra high density. So, I'm kind of giving the game away with my second talk, but ultra high density temporal information that we couldn't address in this competition, I think, is really important, and I'm really interested to see what the other speakers have to say. Okay.
Other question? Thanks again. So now I will speak about the results. We are going to the challenge and we expect to have the results. So my talk is now about the result of the challenge, of course. We expect all of that. So I just want first to remind the goal of the challenge. So there is several minds. So for the developer, it's to try to create a sort of platform to exchange, to boost the innovation in terms of algorithm, and so also to identify the community, because the people do not know the other developer. And also is to force to have to encourage people to have good practice in terms of software design, typically open source, usability, user interface, and so on. And for the end user, it's also really important to create this kind of comprehensive review of all existing solutions and to create a sort of useful resource for end users. And we take the opportunity of this challenge to create some kind of reference common data sets, to try to find good metrics to judge the performance of the software, and to try to go in some standardization way. So the roadmap of the presentation is following. So I will show you the data set that we have. I will speak about the participation, the assessment, and finally I will show the results. And the analysis of the results will be summarized in the roundup talk in Shellus next Tuesday. So do not miss this talk. So the data set. So as Shellus say, we have two kinds of structure. We have microtubule and endoplasmic reticulum. It's all simulation. So we know the ground truth, of course. So we simulate the data in 4 modalities: 2D, astigmatis, double helix, and biplane. You have the 2 plane. In biplane, you have 2 planes, of course. Okay, the 2D focus planes. So as far as I know, it's maybe the first time that we simulate the same structure in different modalities. So which is very interesting to compare modalities. So we create 8 categories, in fact. So we have 4 modalities. Then we have 2 levels of density. So low density, it's around about, I always speak in terms of density in terms of fluorophore per frame. Okay, so it's about 2 fluorophore per frame in average, and high density for us it's about 15 fluorophore per frames. So we have this data. So we have always 2 data sets per categories. One which is with an SNR, with a high SNR, and one with a low SNR. Typically we have 2. In low density we have this ER1, which is a high SNR, and NT2, which is low SNR. So always two data sets per categories. So and 8 categories. So in terms of participation, so we are really happy because we attract the attention of a lot of groups. I think now we have, in end of July, we have 31 software, but we have few in the pipeline. So we have 87 software was run by the author, were run by the author. 61 run at least one 3D modality, which is really cool, nice. And we have, compared to the challenge in 2013, we have 65% of software, which is not, in fact, new competition. So I'd like to thank 3 software, so 3D, WTM, MiaTools, and SMAP, because they did the challenge for all the modalities. So that's very good for them, and probably they adapt their software for the challenge. So the byproduct of the challenge is to encourage multimodalities, so it's very important also in 3D to promote the open source and to foster usability. So that's a huge list of the software. So you can see also this list on the website, of course. So you see in which category they participate. The name in bold, it's not the winner, it's just the fact that the people are in the room. So if you want to know more about the software, you have to you can see that there is a lot of developers right now in these roles. So we check a little bit, we analyze a little bit what kind of algorithm they use inside. So if we took the usual paradigm of the localization, so you have first the detection algorithm, then the localization algorithm, and an important component is also the kind of PSF model they use it. In terms of detection, very often people use it still a threshold or threshold plus filtering. That's the most common detection algorithm that people use it. In terms of PSF model, okay, there is a lot of people that use Gaussian or Gaussian-like models. And in terms of localization, so there is this more various approach. One which is very popular is this MLE, maximum likelihood, or least square approach. Okay, so in terms of assessments now, so we took, so it's a huge work. We collect all the localization file of the participants. So I think there was more than 300, probably we have a lot of more than 300 localization file. We have the localization file, and we have also the calibration file. The fact that we calibrate for the 3D on some bits that we provide to the participants. So the first task for us is to standardize these files, because they do not have the same format. Typically, a classical one is the shift of the origin. There is several conventions to define the origin. So is it in half of the first pixel, or is it in the top corner of the first pixel? There is different convention, and we have to try to align all this data. So it's a huge task. So we had also some control files. So we had the ground truth. We expect a very good result for the ground truth if we compare the ground truth. We had also a software which is called baseline, and we'll speak a little bit later, and also the random, so we generate random localization, and we expect to have very bad results. So finally, we have this huge machinery. We run around all this file, and we have more than 100 metrics. And we produce a lot of tables, plots, images, graphs, and so on. So that we give we opened a few days ago on the web, so it's very useful for the developer. And we also open for the user, such a way that the user can know the performance of the software based on these metrics. This table is now used for making a ranking, because it's a challenge. At the end, we have to rank the software, but it will be used later on for analysis, for a deep analysis. So that's more interesting in terms of science. What kind of performance the software can achieve. So I just record my screen here yesterday to show you what kind of results we have on the web. So we have a leaderboard here, and we can present the software. Typically, we can order like the Jacka index. Here, the ground truth GT obtained 100%, so it's obvious. And the best one is now SMFit here. We can also present the result software by software, modality by modality, so it's a lot again. And we produce all these numbers, so we have the classical one, which is the SNR, FRC, Jacka index. So we have also a comparison based on the molecule-wise, because all of the rest is frame-wise. And we produce images. We compare also in the ground truth and the result of the software. And we have also these nice side-by-side graphs. So there we have two columns and two software. And typically we can show all these graphs, all these images. So there I just extract one example of which is very interesting for the participants, I expect. So we have a systematic assessment of the performance. So we can compare the software to other software, but we can compare also to ground truth. So it's, as I said, useful source of information for the developer. So here I compare software 1 and software 2. In green you have the ground truth, and it's the fluorophore counts per photon number. So typically, if there is a low level of photon number, the software cannot detect anything, and if there is high number of photon number, the software is close to the ground truth, which is really expected. And here we see that this software is very far from the ground truth typically, okay, for the same modalities. So now in terms of ranking, so we select some important metrics, and we have always, we cannot use all the metrics because a lot of correlate between them. So we select a few metrics. So we use this efficiency. So efficiency, it's a comparison between the JACA and the root, the precision, the root mean square, the MSC. So we have now, because we are in 3D, we have the MSC, the lateral MSC, and the axial MSC for the 3D modalities. We also measure the resolution with the Fourier ring correlation in 2D, and in 3D we measure it with the Fourier shell correlation, thanks to Bernd Trigger teams that provide all the code for that. And okay, we would like also to know what kind of range, what kind of Z range is good for a software. So is a software has a good performance in which kind of range? So that's we call the consolidated Z range. So we measure the maximum recall, it's called M, and we multiply by the full width of maximum. So that gives an indication of how good is a software in a range around the focus plane. So I just illustrate a few things. So here a dot is a software, a color is the modalities, and in left you have the low density. So the JACA index here and the root mean square in lateral. So we see that a very good software can achieve 85% in JACA index and with a resolution of 25 nanometer, with a RMSE of 25 nanometer. So in high density, of course, JACA index is low and RMSE lateral is also a little bit higher. And 2D better perform in this case. We have also the axial efficiency, the same thing. So this time we plot the RMSE axial here. So in low density, also we have the same JACA index, but we can achieve very good RMSE axials and less in high density. So I have a few examples, a few images. There we are able to plot the localization ground truth versus the tested software. So in green, in red here you have the ground truth. So this software is not able to detect in border, it is example. And in green you have the tested software. So those are examples, some details, and yeah, then we can produce also these videos because we have. Here in 3D, you have the three microtubules and you can see there that this software has also problems in this when it's very deep. Okay, the baseline performance. So we also develop a software which is called baseline. It's just we have the objective to have the most simple, as we write the code as simple as possible. So, but for the whole modality. So the goal is to know what is the baseline performance. So we did that for 2D estimated biplane DH, and we have also this calibration in 3D. So now the grading is we follow the following process. So we assign the grade 0 to the random, 1 to the baseline, and 5 for the ground truth. So ground truth is always perfect. The scoring is the following. So there is a scoring lateral. So it's the JACA index plus the RMSE lateral plus the FRC for your ring correlation. We have always 2 data sets per categories. And we have a scoring axial, which is this Z range, RMSE axial, and FSC. So that gives two score. And for the 2D, we only use the score lateral plus a little bit point for the computation time. And for the 3D, we have the scoring lateral plus the scoring axial plus point for the computation time. So in 2D, that gives 33 points, the total for the ground truth typically, and 66 points for 3D modalities. So now I can give the result because we rank everything. So I want to to have too few nodes. So first, we do not have the same number of participants for all category. Of course, we have a lot of more in 2D. Some software have several instances, so they submit several files with different settings. So we only select the main one for the ranking, to be fair. Okay. And also, I should say that some participants are in the committee, but they do not have access to the ground truth. There is only 2 people that have access to the ground truth: is me and an assistant, which is Tan Am. Okay. But first, okay, I will announce the result, but I would like to first make some acknowledgement. So I should say that when we start a project like that, it's more than a scientific project. It's like a human adventure, I should say. So we're starting this project 1 year ago with Seamus, and we work a lot together, and it was very, very cool to have all this biophysics expertise. And there is a lot of, always a lot of question when we build a data set, when we build a matrix. Also, we don't know in advance how many participants we have. Maybe we do not have any participants, so that's okay. That's always an adventure. So we have thousands of email, line of code, images, web page, and so on. But at the end, I think we have a really cool feedback from the community, and we expect to have more during these meetings. So there is. I cannot thanks every people, but I just grab all the people that I communicate by email. There is a huge list of hundreds of people that I interact during this challenge. But I would like to have I have special thanks for Suliana Manley and Sandra Radenovic for hosting this event. Thank you, and for Ellen Chang, which is just behind. I would like also to thank the director of my lab, the Biomedical Imaging Group, who is Michael Unzer. And special thanks also to the people that participate to the discussion over the forum. So they are completely volunteer. So Alex Herbert, Joanna Thries, and Hamraj Velmurugan, who participate on the discussion. I would like also to thank Anurag Agarwal, who provide the double helix PSF. And there was a special thanks to Tana Ampan, which is in the room, I do not see. Who is it? No. Here. Okay, he did all the assessments. So that's the guy that has this huge machinery to put all the files and so on, and to come up with all this figure. Without him, it was not possible to do this kind of things. And thanks also to the committee and to the sponsor. Okay. So now it's time to announce the results. So it will be category by category. So, and we have prize, but I just announce first all the result, and then we give the prize just right after. So for this category, it's 3D Dawstorm, so it's a Babcock that have the good result in 2D. So we are in 2D low density, okay? And second is SM-Fold, and PeakFit is the third. In this category, 2D HD, so 2D high density. So the software that's won, that it was GDSC. It's from Stanford. I think we do not have the people here. 3D Dawstorm perform well, and SMFit also perform well. I should say that SMFit is also based on this ADCG. So it seems that this kind of algorithm, astigmatism low density, we have C-splines, again as a Babcock, SM4 was second, TB storm third. Okay. Astigmatism high density, okay, this time it's not Azen, you have only the second one. So it's SM-Fold, 3D Dawstorm, and C-splines. For the biplane low density, so we have Nia Tools, SMAP, and Thunderstorm. For the biplane high density, we have Thunderstorm, Rainstorm, and SMAP. Okay, I should say. For the DH double helix low density, we have C-spline, again, Hazen Babcock. Nia Tools is the DH first. And for the double helix high density, again, C-spline, SMAP, and 3D WT. Okay, that's all the 8 categories. So I have summarized everything here. So that's the first winner of the challenge per categories. So maybe we have some gifts, and I will call the winner. So I think Ethan Babcock, you can come first. Because Ethan Babcock won with, he has two software. He has 3D Storm and C-Spline. And you see very often Ethan Babcock here. 1, 2, 3, 4 categories. Thank you very much for all your participation, and specifically because you participate a lot into this discussion, also to contribute to the building of the challenge. Thank you very much. So, is there someone of ADCG in the rooms? Nick Boyd, Stanford University. So if there is, I think he's not here in this conference, so we will of course send the prize to him. We will not keep it. After I can call somebody from Small Food. I think there is somebody, yes. I'm sorry, I do not know your name, but you will see. I think you're coming from Estonia, is it right? Yes. Just would like to have your name, or maybe Okay, but you can find information on the website.
Yes, I have a poster.
Okay, you have a poster, so more information on the poster. Thank you very much. After from Mia Tools, do we have somebody, maybe Raymond Ober, somebody in this team? Yes. Thank you very much. So after we have Thunderstorm, I think it's Guy Hagen. Yes. And that's it for the prize. Thank you very much for everybody. Thank you very much. Did I miss? No, I missed someone. No, it's okay. No, I missed. Okay, DCG. That's good for you. I see a packet here. Okay. So we are completely on time. We are completely on time to start the roundtables. So I give maybe the microphone to Seamus. So we have 7 experts. They know. So maybe you can sit down here.
Okay. I'll grab the
standardizations that is most important is at the level of understanding resolution and quality of the data. And there's no real good consensus about how to calculate this. If it's detecting the distance between peaks on microtubules, if it's using FRC, or etc. And I think many algorithms will rank differently depending on the metrics that you're using. So again, that can lead either to bias or to ways that you can always find a way to an algorithm to rank better than that one. So actually finding a consensus on the community becomes quite important, which also comes back into the context, because it becomes good if we can somehow update our results in the context, or if someone can simulate at any moment their own tables into the context and see how it's ranking, so that anyone can verify that how different algorithms or different methods play along with it.
So thanks for starting, Ricardo. I mean, I think that standardization is very important. I feel that most of these tools are developed mostly to be used in application to questions potentially not in the field of the people who actually generate them in the first place. So it's a real interdisciplinary exercise. It's quite obvious that the sort of methodology, the mathematics behind how they all work is very different. So actually insisting on a very standard sort of input or way of processing won't work. Potentially looking at the way of having a standardized output. So when you're looking at sort of a longitudinal study, you can actually compare and hopefully reproduce some of the work that's done. I know as a PI myself, I have a small group and I tend to pass projects between students. So actually standardisation is very important for me as a researcher, because what I want to see is that the first student who started it can then pass the work on successfully to the next student and actually get a standardised output. I think consistency within science and robustness is really important.
Do any of the audience want to chip in with a question on this topic at this point? Yeah.
Right. Okay.
I'll just make a comment is that depending on what the end application is, what is the highest priority might change. So say someone wants to use localization microscopy to do high throughput DNA sequencing or something like that, and they just want very high density. They actually don't need really high localization precision. They just need good enough, but they need speed. So, and they need, certain things they need to be accurate. So that's just something to keep in mind in terms of standardization. Depends what the problem to be solved is.
I would agree, but I think what I would say when I put my cell biologist hat on is that sometimes the studies that I do might initially involve something that is fairly high throughput, but then I may find something interesting in that study and then want to follow that up by some very detailed analysis. Maybe, in case of transcription, okay, so I identify a gene which sort of changes the way the transcriptional complex is formed. So first of all, I need something high throughput that just gets me through all the proteins involved. But then when I find the one that I'm interested in, I then need to really zoom in and get something really detailed. And I need to be able to cross-correlate the data from figure 1 with figure 5. And it's a question of how that's then done. How can I share that with my collaborators? I work with some of the people on the panel, and it's great to be able to find ways that we can pass data between ourselves as well in a way that's meaningful.
Do we have any other points anyone wants to make on the topic of standardization?
It's working. Yes, I'm also a participant with Amiro, so it's an open microscopy environment, so they standardize reading of image files. And there was a big discussion on the Amiro forum about standardizing the localization format, and then it tailed off because someone said that there was a publication came out this year, but I can't remember what it was, so I'm sorry for that. But there was an attempt by someone to produce a standard format, and I think that needs to be looked at again, because if everyone who produces software can commit to either using a library in their language that reads and writes that standard format, it makes it very easy for an end user to use lots of people's different software for different analysis tasks, because you can pass around the same data.
Okay, I think OB is quite a good point to stop on, actually, as a maybe topic for future discussion. So next up is the bigger package, right? So this challenge was really focused on the algorithms and how good can they get, but that's not the whole problem of localization microscopy software, right? And so there's clustering, visualization, analysis, and I thought maybe because 3 of the roundtable members actually explicitly discussed the side of the problem and packages dealing with this sort of thing. So maybe Jonas, do you want to comment on this first? What's your view on how to deal with the whole problem?
Yeah, in my group, the main problem is now to get the biological results. It's not so much to get the fit, and then we have the localizations. And I think generally there are 2 approaches: either some kind of probabilistic description, like the cluster analysis and so on, or some geometric analysis, which can either be on the point clouds or on the reconstructed